Hello, everyone. Welcome to our 9-step seminar. I'm very happy to introduce our new seminar with David Deamer, research professor of biomolecular engineering from University of California, Santa Cruz. First of all, thanks so much for accepting our invitation. And the professor's seminar topic will be From Academy to Industry, Evolution of Nanopore Sequencing. We can start if you want.
Okay, thank you very much for the invitation. I'm happy to be here. I've given a couple of other talks now over Zoom to student groups in Turkey. I must also say that I had a Turkish undergraduate student named Ece Kilinç in my laboratory last summer for a 2-month internship. She's from Bilkent University. We also have a Turkish professor named Fitnat Yildiz in the microbiology department here. And Ece worked with Fitnat in her laboratory. So there's a lot of nice communication now between Turkish universities, and our campus. So I want you to keep that in mind as you get a little farther in your career. When you need to think about a place to go next, if you get into graduate school, you could do far worse than to come to one of the campuses in the United States, and particularly up and down the East Coast and West Coast. We have a lot of international students come into our laboratories. So I'm assuming that everybody who is listening is interested in science and has perhaps a science major of some sort. And you've probably had a little bit of chemistry or biochemistry or microbiology. And it doesn't take much more than that to understand the talk that I'm going to give. So there's really two kinds of science that you can get into as a career. One is called basic research, and that usually goes on in an academic laboratory. In the US anyway, we have quite a bit of government support, and most of our science faculty have grants, and most of the faculty on my campus have 2 or 3, 4, 5 grad students or postdoctoral associates in our labs being supported by science grants from National Institutes of Health, from the National Science Foundation, and in my case, from NASA. So I get money to study the origin of life. And that is how this idea that I'm going to tell you about, how it began. And what you're going to hear is how an idea that began in a basic research setting turned into an industry that is now global. And it's been wonderful for me to watch the growth of that from an idea to a major industry related to sequencing genomic information, in particular sequencing DNA. So I'm going to start my slides now. So there's the title, From Academe to Industry, Evolution of an Idea. And I'm going to tell you 12 lessons that I have learned over this 30-year period as an idea got picked up by a startup company and who then successfully got it all the way to the market with scientific instruments that you'll hear about. This was an idea that started back in 1989. I'm going to show you my notebook from back then. But first, I'm going to tell you, where do new ideas come from? And a lot of new ideas come from academic settings, not so much from industry. Industry likes to pick up ideas that we bring to them, we faculty members in a science department. And this is an idea that got picked up by a startup company in the United Kingdom. And you're going to hear about that as I go on. So I was on the faculty at the University of California, Davis in 1989, and I was doing basic research trying to assemble artificial life. So that's about as basic as it gets, because nobody much cares about artificial life, just a curiosity, I suppose. But I want you to learn from this that ideas can come out of just curiosity-driven research that then have value to the society that supported them. And the society, in my case, are the American taxpayers, because their money, a tiny bit of it, goes into supporting research at the universities in the form of grants. Now, a grant is money that doesn't have to be given back. It comes to us as a gift. We have to compete for it, and it comes to our university campus, and it becomes part of the accounting that goes on at a university campus. And that money then is, I have control over how it is spent, because I need to support graduate students and postdoctoral students and a technician, for example. And that is most of the grant money that comes to universities is salaries. So not so much supporting the actual research and the equipment that we have to buy and the supplies, but it does support about 80% of it goes to our grad students and postdoctoral students. So I was trying to figure out how to make life in the laboratory. And I'm still trying to figure that out. 30 years later, it's been a lifelong interest of mine. How did life start? And can I get this to work in a laboratory situation? So the recipe of the materials and compounds that I was using were lipid membranes. And maybe you've heard of this. These are called liposomes. And they were invented by Alec Bangham in the 1960s, who worked in England. And I did a sabbatical with Alec years ago in the mid-1970s, and that's where I learned about lipid membranes and liposomes from the person who discovered them. So that is another thing that you do in an academic career. You are allowed to go on sabbatical leaves for 6 months, in my case, to a year, sometimes a little bit longer, and you earn the privilege of going on a sabbatical by teaching and doing research at your university campus. So that is an established thing in the United States, that most of our research universities allow you to go and your salary continues. In other words, you are paid to go someplace else in the world. And in my case, I went to a little village near Cambridge, England, called Babraham. And that's where Alec Bangham worked at the Animal Physiology Institute. So that's how I got interested in lipid membranes. I also was interested in ion channels, because that's how life works. All cellular life has ion channels across the membrane. So I knew about ion channels. And in fact, in my research, I was working with an ion channel called gramicidin, which is an antibiotic. In fact, if you buy an antibiotic salve that you put on if you injure yourself, you would buy neosporin or polysporin, and that has gramicidin in it. That's one of the compounds that fights bacteria. So we were trying to figure out how that worked. So that's my introduction to ion channels. That's what I was doing. We also used electrophoresis in the laboratory. And you've probably heard of this. This is a way to make charged molecules move through a gel. So these molecules, such as DNA and RNA, will move through a gel that has nanoscopic pores in it. It's like a mesh. And these long, stringy molecules of nucleic acids when they feel a voltage that we impose across the membrane, they will move through the gel. Fast molecules are the small ones. They move fastest. The large molecules take longer to find their way through the gel. And then finally, I knew about Coulter counters. Now, you may not have heard of Coulter counters, but if you have a problem and have to go to the hospital, they will take blood from your vein. And some of you probably have had that happen to you. And they put it into something like a Coulter counter that counts your red cells and your white cells. And I knew how to do that. So those four things were in my head as an unconnected knowledge. And I then accidentally made a connection that turned into this idea I'm going to talk about. I needed to get ATP into liposomes containing an enzyme called RNA polymerase. Now, all nucleic acids in life are made by enzymes, and RNA polymerase takes things like ATP, adenosine triphosphate, this is the energy source of all life on Earth, and CTP, cytosine triphosphate, and guanosine triphosphate, and thymine triphosphate, and so forth. And these are the four bases of DNA. So those four bases contain the information when they're in the sequence of bases in a strand of DNA. That's how a cell knows how to make a protein. And you probably know that from your introductory courses that you have had, that DNA has the genetic information that guides protein synthesis. So I wanted to make this enzyme, RNA polymerase, synthesize RNA using things like ATP. And that was just the beginning of this idea. Well, there it is. That's a problem because membranes are a barrier to ATP. ATP cannot get into a liposome. It's too big. So I was looking around for an ion channel that would allow ATP to get into these membranes. I had a postdoc in the lab named Mark Akerson, and this was now 30 years ago, more than 30 years ago, and he was in my lab as a postdoc. He then went off to NIH, the National Institutes of Health, but I invited him back to UC Santa Cruz when I got started on working with this idea that I'm going to tell you about. So he's one of the names that contributed to all of this technology that turned into nanopore sequencing. Dan Branton was my postdoctoral advisor while I was at the University of California, Berkeley. So isn't that interesting that there's three generations? There's Dan Branton, who was my advisor, and Mark Akerson, who I was a postdoctoral advisor when he was at UC Davis. So he got a job with us. He did such wonderful work that he was hired as a professor in my department, biomolecular engineering, here at UC Santa Cruz. Okay, now I'm going to show you this idea as it developed. Here's the liposome membrane, that circle, and there's the enzymes. The round balls are that's the enzymes that are going to work with ATP to make RNA. And you notice that ATP cannot get through the lipid bilayer of the membrane. So that's a problem for me. So I thought to myself, maybe I could find a protein channel that would let ATP that I add to the solution get into the liposome and turn into RNA by the enzyme. So I was thinking about what kind of a protein channel would allow ATP to get in. So then I thought to myself, wait a minute, if I can put a voltage across the membrane, I could pull the negatively charged ATP from the negative side of the membrane to the positive side of the membrane. I could literally pull it across the channel by imposing a voltage. And then it occurred to me, this is like a molecular Coulter counter. Maybe when the ATP goes through the channel, I will see a blockage of the ionic current, that is also going through the channel. Then finally, the idea popped into my head. What if this ATP is not all by itself, but is part of DNA? Maybe each base in the DNA, as it goes through the channel, will block a different amount of ionic current. Then, as it goes through the channel, you can see down at the bottom on the right, you will see different levels of ionic current blockades. And they're just little millisecond downward spikes of ionic current as this piece of DNA goes through the channel. So that was the basic idea. Now then, here is lesson number 1. Write down your idea. I had this idea while I was driving in Oregon. I had been to a meeting in Eugene, Oregon, which is where the University of Oregon is. And I was driving east to the Belknap Lodge, if you can read my handwriting, where my family was on vacation. That was in eastern Oregon. And all of these ideas that I told you about were just sort of tumbling around in my head. And I realized that I might be able to sequence DNA by pulling it through a pore. And you can see my writing about this. Off to the left, it says main concept. DNA will be drawn through a small channel, either by delta psi, that's actually the membrane potential, that's the voltage, or delta pH. Now, we never got around to trying delta pH, but we do use the membrane voltage. The channel will be carrying an ionic current driven by a voltage. As each base passes through, a change in the current will occur. Because the bases are of different size, the current charge will be proportional, thereby providing an indication of which base it is. Then off to the right of this notebook, you can see a little strand of DNA going from right to left. It's being, from left to right. It's being pulled through the channel by the applied voltage, which is positive on the right-hand side. So the negatively charged base will be pulled through. And as each base goes through the channel, down at the bottom on the right, you'll see I, that's just ionic current. And you can see these individual blockades, C for cytosine, G for guanine, T for thymine, and A for adenine. And that was the basic idea. Now remember that, because you're going to see it again in just a little bit. So here's lesson number 2. Patent your idea. So if you have an idea that you think might have value in society, or for a company that might want to use your idea, you have to get a patent. So I had a friend come to visit me. His name is Daniel Branton, and you can see his name in this little list of inventors off to the right. George Church, myself, Daniel Branton, Richard Newt Baldarelli was the postdoc in George Church's lab, and John Kasianowicz was the person who had a channel that we wanted to use. So those are all the inventors on this grant, on this patent. And you can see that the patent is owned by the president and fellows of Harvard College. So the inventors in an academic setting don't own their ideas when they get patented. It's owned by the university, but the inventors get a share of any royalties that come from the patents being licensed or purchased by a company. So that's how the inventors make a little money off of it. So that is sort of, that helps you say, hey, I might be able to use this idea and get a little extra money out of it. So that's a good reason to be an inventor on a patent. In the case of the University of California, we get 1/3 of all the royalties that come from a patent that is licensed by a company. So that's simply a negotiated share that inventors get. Okay, off to the left now, you see the title of this patent: Characterization of individual polymer molecules based on monomer interface interactions. And you notice that it's not called a nanopore. The patent was issued before we invented the term nanopore. And you're going to see where that invented term was first published in 1999. So you can see the patent is 1998. When it was published and issued. So that's the way it works. You really do have to patent your idea, because otherwise a company will not be interested. They need to have ownership of your idea. And as an inventor, you allow the university to license your idea by a company who might want to develop it. Okay, so that's how an idea is transferred from the university setting to an industrial setting. It's called technology transfer. And on most university campuses, there's an office that is responsible for transferring your ideas to industry in the form of a patent. So keep that in mind as you go farther in your career. Patents can become valuable, both to society and to you as an inventor. Well, we just had an idea at that point. We applied for patent in 1992, and George Church had a similar idea. He was also at Harvard, and we simply decided, we don't know if this idea is going to work, so let's all be inventors, listed as inventors, because we have no idea whether it's actually going to work or not. So that was really very nice, because that's the way colleagues work together. They share their information, they share their ideas, so everybody gets involved. And that expands the number of people that might be able to help it work. So John Kasianowicz at NIST in Maryland, the state of Maryland, that's called the National Institute for Science and Technology, John had a channel in a membrane called hemolysin, and you can see it on the left there. And I looked at that and I said, that channel might be big enough to accommodate a piece of DNA, a single strand of DNA going through. And then on the right-hand side, you can see a strand of DNA being pulled through that channel by a positive charge, a membrane potential. It's about 120 millivolts. It's not very large electrical voltage, but that does pull it through. So that was an idea. In 1993, I traveled to Maryland, and John set up his channel for me, and we added some RNA to the solution in which this bilayer with the single channel was prepared. Okay, now I'm going to show you what we saw. We saw hundreds of downward deflections of the open channel current. That's the 120 picoamps. PA stands for picoamperes. And that's not a very big current, by the way. That's, that's what, one trillionth of an amp, I guess. A nanoamp is 1/1000th of an amp. A microamp is, about what goes through an ion channel in our bodies. So that's not a very big current. But as the DNA goes through, and you can see the open channel current with the red and the blue arrows on the left, that's the 120 that you can see. And then as soon as the DNA enters the pore, it blocks the current for a few hundred microseconds as it zips through. And then it goes in and out. And each of those downward deflections is one molecule of DNA or RNA, in this case, going through the channel. So we knew that this might work. If that experiment had not worked, we would have just given up. It's just another idea that didn't work. But it did work. So we got very excited, and that's when we really got to work on this and went for a patent. So this is 1993, and this was part of our patent, which was issued in 1998. So we call that translocation, and either DNA or RNA will go through the channel. Well, it's a secret. You don't tell anybody about that until you publish. So that's lesson number 4. Suddenly, after you started your patent application, you don't do that. You don't publish before, because if you publish before, you won't be able to patent your idea. You've already shared it with everyone. So you keep that idea confidential until finally you know that you're going to be able to patent it. And by 1996, we knew that we would be able to publish some of our results and then use that for the patent. So look at that big red arrow down to the bottom of this, of the abstract. And I'm going to read the abstract to you. With further improvements, the method could, in principle, provide direct high-speed detection of the sequence of bases in single molecules of DNA or RNA. So that's when we revealed this secret idea to everybody that wanted to read this paper. People were very skeptical. We had a lot of trouble getting grants because this seemed impossible. How could we possibly pull a piece of DNA through a single molecule and see each base produce a change in the ionic current? Very skeptical. That was a problem that we had. We sent this paper to Nature. It was declined. We sent this paper to Science. It was declined because nobody could believe it. We finally sent it to the Proceedings of the National Academy of Sciences. Dan Branton is a member of the Academy, and therefore we finally got it into publication. And you can see, contributed by Daniel Branton, September 5th, 1996. So because of his membership, we got this wild idea published. This paper has now been cited close to 4,000 times in the scientific literature. And it just shows what can happen to a really good idea that your colleagues are skeptical about. They're gonna wait and make you wait until you get it published. You gotta have more sort of evidence before people really start to believe it. So we needed more evidence. So here it is. In 1999, we published another paper. So there's now 2 papers in the literature claiming that maybe we can do this. And what we said is that we could actually watch in a single piece of RNA, we could watch a pyrimidine polymer, the polycytidilic acid, and a purine polymer, the polyadenilic acid, in the same molecule. We could watch it go through and see a difference between the purine and the pyrimidine polymer. And also in the abstract, I'm going to put a little red square around a new word. Do you see that down there? That's the first time the word nanopore was used in a published paper to indicate a protein channel that can let a piece of nucleic acid go through. So it's kind of fun to realize that nanopore sequencing started with this paper and all the papers that followed. Well, we had to license our patents, otherwise industry would not be interested. So in 2007, a startup company called Oxford Nanopore Technologies, the CEO and the CFO, that's the Chief Executive Officer and the Chief Financial Officer, Gordon Sangara and Spike Wilcox, came to visit us at the UC Santa Cruz campus. And they said, we want to license all of the patents that you've developed. And that's called intellectual property. And they now have licensed 1,000 or more patents to protect their company. So we said, well, sure, let's work together on this. The university, of course, owns the patent. So the university actually negotiated with these two guys and their company how much the university would get in the way of dollars for licensing their patent. And it wasn't terribly expensive. They had access to something like 100 million pounds to support their company. And this is the licensing fee is a relatively small amount, just a few $100,000, for example. So these were the guys that we went to industry, and you have to license your patent, and they came to us. And that's unusual. Usually you have to sell your patent to a venture capitalist, it's called, where they provide the money. And it's very competitive to get the millions of dollars it's going to take to develop your idea. But here, this company came to us, and that was really good because finally somebody in industry was interested in nanopore sequencing. We had a little problem though. Hemolysin wasn't going to work for us. So you can see a piece of DNA in alpha hemolysin in the green blob on the right. And you notice the sensing region. That's about 10 bases in it. All 10 of those actually give the blockade of the ionic current. They contribute to it. So how can we get base-specific blockade if 10 are contributing to the ionic blockade? So a colleague of ours at the University of Washington named Jens Gunlock came to visit us, and he said, I'm interested in helping with nanopore sequencing. And I want to work with you guys. I'm going to learn how to do it. So we showed him how to do channels in a membrane because we'd gotten pretty good at it. And he went back to his campus and he found a new pore called MSPA, and that's the one on the left. And instead of being a mushroom shape with a stem, it was a funnel with a much smaller sensing region that could sense only a few bases at a time instead of 10 or so. That really caught everyone's attention. He published that paper in 2010. We were all getting support, by the way, by that time from the National Institutes of Health. They had a program called the $1,000 Genome. And our aim was to get a human genome for $1,000 instead of millions of dollars that it took at the time that we got started. So we all sort of went over to MSPA, and that included our company. And we then did a little bit more work, and we were able to sequence DNA by combining a polymerase that we had developed with the MSPA channel that the University of Washington people had developed. So lesson 7 is to publish your article in a top journal in Nature Biotechnology. And I'm going to show you what we published. Oh, by the way, you can see how this works. That little single strand that goes through the pore is being pulled up through the sensing region of the pore because it is being turned into double-stranded RNA by the polymerase. That's a Phi-29 enzyme polymerase from the Phi-29 phage. It's a bacteriophage that happens to have this enzyme in it. So as it makes DNA at about 100 bases per second, the single strand gets pulled up through the pore, and we can then watch it be ratcheted through the pore. And this is what it looked like. This is the first nanopore sequence of DNA. So notice that this is a synthetic DNA that has ACT, ACT, ACT. Then we put in a G just to see what would happen. And then it goes back to ACT, ACT. And you notice that A starts up at the top, and then C is in the middle, and T is down at the bottom. Then notice top, middle, bottom is T. Top, middle, bottom. And look how that compares to the original sketched idea. That little dream of mine was made real by watching the nanopore sequence a real piece of DNA. Meanwhile, the company was hard at work, and they did something that we could not do. And they announced it at this meeting in Florida at Marco Island, and is the AGBT meeting, Advances in Genome Biology and Technology. So we announced our ability to sequence DNA, and Clive Brown announced that the company was going to be able to put out an instrument that could use nanopore sequencing, and anybody would be able to buy the instrument. So you're going to see what that looks like. These are the people that are involved in the academic side. Jens Gundlach is clear to the left. One of the women from the company, from Oxford Nanopore, is there in the black outfit. Mark Akerson is next. He's in the center. He was my original postdoc and then colleague as we developed all this. I'm just sitting there looking up as somebody's saying something. And then Dan Branton is sitting right to my left. And that's the group that really did the academic production, the academic achievement. But at the same meeting, Clive Brown said, we are using nanopore sequencing in an instrument that we're going to distribute. And now you're going to hear the company story, because the company, with several hundred employees and several $100 million of money to develop it, can do something that academics cannot do. Our device is 1 nanopore, and you can see the nanopore there in a lipid bilayer. The company's device is 2,000 nanopores in a little chip called ASIC. If you look down at the bottom, you'll see application-specific integrated circuit, ASIC, an ASIC, and that is built into a sensor well that has 2,000 nanopores in it. Every nanopore has DNA going through it, and you simply plug it into a laptop computer and away you go. This could be purchased for $1,000 compared to $100,000 for the nearest industrial competing device. This is called disruptive technology, and it's dangerous to come out because this little startup company is competing with industries that are worth billions of dollars. And our little company, Oxford Nanopore, had maybe 100 million pounds to work with. So you're going to learn something in the next lessons. So lesson 10 is to do something no one else can do. This is on the International Space Station, And that's Kate Rubin. She's an astronaut, and she's floating in microgravity in 2016, not too long ago. And she's using a minion to sequence DNA in the International Space Station. So down to the lower right, you can see the minion. It's plugged into a laptop. It's going up to that screen, and that screen shows an indication of the numbers of molecules of DNA that are going through it. So that is fantastic. That no other instrument is small enough to go up to the International Space Station. They weigh close to 100 pounds. This little guy is the size of a cell phone. So that is really wonderful advertising. It is also was used in the Ebola epidemic. Nick Lohman and Joshua Quick went off to Africa. And as the Ebola was going from village to village, they were able to use the minion to sequence the DNA of the virus and identify what the virus, how it was spreading through the villages. So that was really something. That caught a lot of attention. Finally, people believed that nanopore sequencing was going to work. So we then learned a couple of hard lessons. And that is when small companies invent disruptive technology, big companies sue. Big companies don't like it if somebody undercuts them in the market and sells something for $1,000 that they have for sale for $100,000. So Illumina sued Oxford Nanopore for patent infringement. Do you remember that MSPA pore I showed you that was being used? Illumina claimed that Oxford was using that nanopore, which they had licensed from the University of Washington. We had to go to court. One of our faculty members was an expert witness in the trial. You can read about it. That's just the patents that were involved in it. They claimed that we were infringing their patent, but we won. We won the suit, and the lawsuit was won because Clive Brown, the chief technology officer at ONT, said on a public lecture, "No thanks, we already have one." And that is a quote from a Monty Python movie called Monty Python and the Holy Grail. And there it was a French soldier turning the British knights away, saying, "We already have a Holy Grail." Well, Clive Brown and his people at ONT had gotten their own pore, and they said, "We don't need MSPA for our devices." And so they won the lawsuit, and Illumina had to settle. So that's the first lawsuit. But guess what? There was another lawsuit. PacBio, another big company, sued Oxford Nanopore for alleged patent infringement. Isn't that amazing? Again, one of our faculty members, Mark Akerson, was an expert witness at this trial, and the jury decided that the PacBio patents were invalid, and we won that one as well. And that was just a couple of years ago now. So you can see what happens when you come out with disruptive technology. At the billion-dollar level, the big companies can afford very expensive lawyers. They can afford the cost of having these lawsuits. They're hoping that the small company won't be able to afford the fight and will have to either just go away or settle. So lesson 12 is, in the face of adversity, carry on and muddle through. And believe me, the British people are good at that. Here is the products that Oxford Nanopore now has for sale. There's the minion to the left. They have a little bit bigger one that has 5 minions in it. That's called the gridiron. And then the Prometheon is clear to the right, and it has the equivalent of 40 minions. The Promethean can do a human genome in 8 hours. It has set a new record for taking a patient with a genetic disease and getting a complete human genome using the Promethean. Stanford doctors have also just very recently used the Promethean to do an 8-hour genome of patients in their care at Stanford Hospital. And that's where that new record came from. And our faculty members were involved in that. So the Promethean is about $125,000. The MinION is still about $1,000. The GridION, I forget what that is. Not so many of those has been sold, but they might be $10,000, $20,000. I forget the exact cost. But that's pretty good. And now here's the last lesson. Finally, Oxford Nanopore had an IPO. That's an initial public offering. And that was last September. They came home with over $4 billion of investors' money. So they are now valued at a $6.8 billion company. And that is 30 years from the time that Dan Branton and I got together in Davis, California in 1991 to talk about getting a patent. So that's a story of what can happen and what has happened before. Another person who lived this story is Jennifer Doudna. She got a Nobel Prize for CRISPR just last year. She was one of the Nobelists. So that's a prize that marks a very successful transfer of technology from an academic to an industry that's now using CRISPR. That was a very valuable patent to go for. So that is my story. And I think what I'll do now is to take any questions that you might have. So I'm going to stop sharing.
Are there any questions you want us to, Professor? You can write it in the chat box, or you can ask after unmuting yourself.
I think you have to do the stop share. I've done it at my end. But I think it has to be turned off at your end to get back to the seeing people.
You're not screen sharing right now.
If you can't get back to it, of course, I can just listen to the questions and answer verbally.
Okay. I can start with the questions which were written in the chat box, if you want. The first question is, in which areas Nanopore sequencing technologies can be used?
Okay, I'm going to have you ask that question again, because it doesn't come over very well over between the computers. So ask it one more time.
Okay. The first question is, in which areas Nanopore sequencing technologies can be used?
Okay, in this area, Nanopore sequencing technology, is that the question? I haven't heard a question yet.
Yeah, the person asks, in which areas the technology can be used?
There we go.
I think there's an internet connection problem.
Yeah. Okay, try that question one more time. I'm sorry if I'm not getting it.
No, it's not a problem. The first question is, in which areas Nanopore sequencing technologies can be used?
Oh, in which area can nanopore technology be used? Is that the question? Got it. Okay. It is being used diagnostically in medicine now. The company would love to have an instrument that they can put into doctors' offices so that a patient can come in. The doctor will take a little bit of a few cells from the cheek, you know how you give for the COVID testing. So those could be then analyzed for the genome, and if maybe, half an hour later, he has genetic information that will tell him which bacterium is producing a disease or which virus is producing a disease or whether there is a genetic disease. So that is the most lucrative application of nanopore sequencing, because everybody goes to the doctor. And if you have a way to very quickly get a viral diagnosis or a bacterial diagnosis or a genetic diagnosis, that would be very valuable. And the company would sell lots of instruments to doctors' offices. And by the way, nanopore sequencing has sequenced about 1/4, 25% of all the COVID viral genomes worldwide. So this is already being used diagnostically to see what a virus is that a patient has.
Okay, thank you so much. I think I can jump to the second question. The second question is, hello, Professor. I want to publish a scientific paper during my bachelor's. What are your suggestions?
Good. You should try to get into that as fast as you can, as soon as you can, because no matter what, whether the paper is accepted or not, it's good exercise and gets you started, because it's a little bit scary sending off a paper that you've written, particularly your very first one, sending it off to a journal to get published, because it's going to go through peer review and very knowledgeable people are going to read your manuscript and they're going to get back to you with either saying this can be accepted, but you have to do this and that, or fix this. You know, they will be constructive, but they may say something like, we don't think this is appropriate for our journal. And so it's a polite way of saying, no thanks, just go away, try some other journal. Now, young investigators have at least one special journal, and one of my students has had her paper published there. And this is a high school student, and we have a college student now also publishing. It's called the Journal for Emerging Investigators, and is published by Emory University, and the editor is a postdoctoral person there who's just doing this because she thought that there really should be an online journal made available for the people to try their first publication. And it does a good job. They send their papers to excellent and sympathetic reviewers. They know it's your first paper, so they try to help you get it up to a published paper. So you can go to a journal like this one, Journal for Emerging Investigators. You can find it online, but there's another way to get published as well. I would say that a beginner could never get a paper published in Nature or Science. In fact, lots of people cannot get their papers published in Nature or Science because it's extremely competitive. It's just, you have to go to the next set of journals down from these major international journals. The next set, survey field, like there is the Journal of Cell Biology, the Journal of Microbiology, the Journal of Biomolecular Engineering, for instance. There's all kinds of journals that are specialty journals that serve a special field rather than Science and Nature which serves the entire field of science. See how that works? So then, even under that middle level, there's a low level of journals that are internet only. So they are published, they're open access, and you have to pay a little money to get published there, but some of them are pretty good. I have been an editor of a journal called Life, and it's a very good journal. A lot of people are publishing there now. It's published by MDPI Publishing, and it's online only, and everybody can read your paper. There's no charge. It's called open access. So those are the areas that you could likely get published in because there's simply less competition to get into these online open access journals, but it costs a little bit of money. You'll have to maybe pay $1,000 to get published there because they have costs associated. So I hope that answers your question on where to publish. If you have a manuscript ready to go, what you can do ahead of time is to let other people read it. And in particular, if English is not your native language, it is the language of science. So you've got to be able to be pretty grammatically correct in your writing. And that's hard to do. If you have grown up, for instance, with Turkish as your mother tongue, it's hard to write clearly, but at least you can try to do it and then get help from your peers, people like I see on screen, or from your faculty member, or you can even pay somebody. There are people that will professionally edit your grammar in your manuscript, but your grammar has to be correct and your spelling has to be correct. And every journal has author's instructions. So before you submit, you must read the instructions that the journal has in terms of how to cite papers, how to organize what you're saying, how to use abbreviations and so on. So it's a pretty steep learning curve, but once you learn how to do it, it gets easier and easier and easier. So try it, put your toe in the water, see if you like it. Okay, other questions?
The other question is, why was Kate performing DNA sequencing in the ISS?
Well, why did they sequence DNA in the International Space Station? The reason is that it's been floating around up there for what, maybe 30 years? I forget exactly when we put it up there, but people have been living there for all that time, and people are full of microorganisms. We have bacteria as part of our bodies. We have fungi as part of our body. We have viruses. We're sharing viruses. The COVID virus is an example of something that we've all shared among ourselves. And those coat the walls of the space station. If an ordinary bacterium happened to get a mutation by just spontaneously or maybe radiation coming into the space station, and if that mutation caused it to become pathogenic, there are problems. You all know, I think probably, that E. coli is a common bacterium in our intestine. It's just everybody's got E. coli that we carry around. But once in a while, there is a mutant in E. coli that produces a pathogenic strain, and that can be fatal. Some of these E. coli strains are absolutely fatal if you come down with it. We don't want that to happen in the space station. So they want to be ready with a device that can give them an indication of what virus it is or what bacterium it is that is causing a disease in one of the astronauts. They want to get that right away. And the Nanopore sequencing device can do that for them. So that's why they have the Nanopore Ninayan up there in the space station? That's a good question.
I cannot see any more questions in the chat box, but if you want to ask anything, you can ask after unmuting yourself or writing in the chat box. I think there is no more questions. By the way,
could I say something?
Yeah,
after this kind of a talk, you have my email address, and often people might not, want to say something right away, but they send me an email, and I'll certainly try to reply. Okay.
Okay, thanks so much for this.
I have a question. Sorry.
You can ask.
What are your suggestions for us to, in terms of following the industry, like as a student, what can we do? Or do you think it's how much it is needed for us, or what can we do, or how much skills is good to have for us?
Okay, I didn't quite get all of that, so try to speak a little bit louder and very simply so that I get your question.
Okay. What are your suggestions for us as students in terms of following the industry? For example, if we are interested in something, what are your suggestions for us to follow the industry?
Okay, I got it.
What skills we need?
So what are my suggestions for you as students if you want to go a little bit farther right now? And the answer is internships. They're by far the best experience you can have, either within your country. You do have industry that loves to have internships, and interns are very inexpensive for a company or for a university, and often you are a volunteer intern, so you're not really going to be costing any money, but you get invaluable experience of what it's like to work in an academic lab or in an industrial setting. So internships are by far the thing that you can do right now, looking around for somebody to take you on for a summer, for example. There are international internships. We have quite a few international students, such as Ece Kilinç, who came to my laboratory. She had her own support, and I'm not sure whether it was government support or just her family was supporting, but she had $6,000 that she could come with, and that paid for her travel, for her local expenses, for the apartment that she was living in on campus. So if you can get an international internship, that's even more fun. You get to see another country and another culture, and you get to practice your English. Very important, to have, because that is the scientific language. So that would be my answer to what you can do, think about right away. Of course, let me tell you about graduate school. I gather you're mostly undergraduates. If you can get into a graduate school in well-funded countries like the US, like Europe, like the UK, like Japan, even China, there are places where you can do graduate work, get a PhD, and a PhD is well worth your investing time because it puts you at another level of earning. With a bachelor's degree, you will basically become a technician in industry if you go into industry, or you become a research assistant in an academic laboratory. So those are not very well-paying jobs. But if you have your PhD, suddenly you're going to get significantly more money out of the investment that you make in terms of time and money. So try to get a PhD if you have a chance. It's a wonderful thing to do. I loved my graduate work. Then after that, there's even a postdoc. If you're going to go into academics, it's really good to have a postdoctoral research. And there you're getting paid to work at some university laboratory and get publications. Because when you really go for a academic position, you're going to have publications of research with your name on the paper, and it's going to have to be your research. And only then would you be able to compete for an academic position at a really good university. Okay?
Thank you so much.
Sure.
Before ending the meeting, you can find the links to the journals Professor Deamer mentioned in the chat box. And we add the mail of the professors in the chat box. Thank you so much, Professor. It was so good to listen and learn from you.
Good. Well, thank you for the invitation again. I've enjoyed telling you about my experience. Bye-bye.
Thank you.
Bye.
Bye-bye.
Thank you.
Great.